we are going to continue. I would like to remember our other audience that you can make your questions in the Q&A box in the bottom of your screen. And also you have the interpretation option there to choose your language. So our next talk, again by Dr. Rebecca Wells, will be about the role of the matrix in fibrosis progression. Thank you, Dr. Wells.
Can you see this now?
Yes.
Okay. Thank you again for the introduction, and thank you for inviting me to give this second talk. I'm going to continue where I left off in the first talk and talk to you about the matrix of the injured fetal extrahepatic bile duct, and suggest to you that we can think of biliary atresia possibly as fetal wound healing gone bad, if you will. And so let me explain to you why we think that that might be the case. So in this talk, I want to cover 4 topics. First, I want to cover briefly the features of fetal as opposed to adult wound healing. I want to talk about how there is increased hyaluronic acid found in the normal and injured fetal extrahepatic bile duct of any species we've looked at, including human. How increases in HA may lead to obstruction, and how HA also has a significant role in stimulating the extrahepatic bile duct cells to grow if they're cholangiocytes and to become fibrogenic if they're fibroblasts. And the model that I'd like to suggest is shown here on the right, where when you have injury, the increased amount of hyaluronic acid may lead to obstruction and other consequences. So first, I would like just to briefly review the features of fetal wound healing. So fetal wound healing has intrigued scientists for a very long time because it seems to be scarless. So particularly early in gestation in humans or most other species, if you carry out surgery on the skin, or for example, there's a myocardial infarction of the heart, the epithelium is regenerated and there is no scar formed. This has been studied in the skin, the heart, the lung, the tendon, and I'm actually showing you on the right-hand side figures of what happens after a myocardial infarction, an induced myocardial infarction in the fetal versus the adult sheep heart. And you can see the fetus on the top left seems to have 4 weeks after infarction, seems to have a completely normal looking muscle, whereas in the adult you can see left ventricular thinning and marked scar tissue, and the stains down below that confirm that there is significant scarring in the adult but none in the fetus. And this, as I said, is shown in skin, but also a number of organs that would be considered much more similar to the bile duct. So features of fetal wound healing include very high levels of hyaluronic acid. They also include very high levels of collagen 3 compared to collagen 1. These are both fibrillar collagens that have a major structural role in the body, but collagen 3 seems to be less scarring than collagen 1 when it's been studied in a variety of contexts. There are also very high levels of IL-10 present, and the overall environment is anti-inflammatory from the perspective of wound healing. And as I said, this seems to be especially true early in gestation. So I just wanted to briefly cover what hyaluronic acid, abbreviated as HA, is, because it's so important in fetal wound healing. And it's a massive glycosaminoglycan or carbohydrate. It's made up of repeats of this pair of N-acetyl D-glucosamine and D-glucuronic acid, which I've shown you here at the bottom. So, I mean, there are just thousands and thousands of repeats of this pair. HA, it's highly charged. And as a result, it attracts massive amounts of water and swells. And if you actually try to look up hyaluronic acid, say on Google, all you will find are cosmetic products, because because it attracts water and swells, it's very attractive for cosmetic uses, but it does highlight, or that point does highlight that HA does attract water and seems to swell. It is originally secreted as a high molecular weight form, and this is up in the megadalton range, but it can be broken down to low molecular weight HA, and that's in the kilodalton range. So there's really a very markedly broad range of sizes of this polymer that you can see in the body. And interestingly enough, the high molecular weight and the low molecular weight have quite different functions. The high molecular weight in general seems to be beneficial, seems to have anti-scarring effects, whereas the low molecular weight seems to be pro-inflammatory and is considered to be more detrimental. So before I go into the data, I just need to introduce a model that we've used for some of these studies. I'm going to show you a lot of work, again, with rodents and some work with human tissue. But this was a model system that we essentially stumbled upon, but has turned out to be very useful for us. So this is the fetal sheep biobag. And we got this tissue from Bill Gaynor and Mallory Hunt at Children's Hospital of Philadelphia. And what this is, it's a pseudo womb that is being tested for use in severely premature infants. In other words, these infants that are born prematurely get put into this biobag, and the thought is that it's the equivalent of a womb to get them to a point later in gestation when it would be healthier to be born effectively. So what is done is that pregnant mother sheep undergo C-section with removal of the fetus, and the fetus is put into the biobag for 7 to 21 days. Control animals are just continue to gestate in the maternal womb as usual. The prenatal, the animals in the biobag, in some cases the biobag is made hypoxic, so they may have in general hypoxic injury. A number of different tissues have been studied in this model, but we've looked at the bile duct. So I just want to, before going into the data on HA, I just want to highlight that one of the interesting features of the fetal bile duct compared to the adult bile duct is that the peribiliary glands, that are these little stem cell containing glands in the submucosa, increase really quite dramatically in number and proliferate in response to injury. So what I'm showing you here is data from the sheep, but I think it holds in other species. And you can see that when there's injury, in this case, hypoxia in the biobag, there's a really dramatic increase in the number of peribiliary glands compared to the controls. This also seems to occur more quickly after injury than in the adult. And I should just say that this work has been done in my lab by Iris de Jong, a graduate student from the Netherlands, who has done quite a bit of work in adult ducks looking at ischemia reperfusion injury. And so many of these comments about fetal and adult PVGs are based on that model of adult injury. They are randomly distributed rather than clustered in the fetus, and they're minimally dilated compared to in the adult. So with that, we looked at both the sheep model as well as normal neonatal and adult mice, and looked at the HA content in the extrahepatic bile duct. And what we saw was there was really a marked increase in the amount of HA present in the fetus and the neonate compared to the adult. And in fact, when Iris went and quantified this, we found that in the rat and mouse neonate, about 40% of the duct by weight was HA. And about in the adult, that went down to about 10 to 20%. So there's really quite a significant increase or significant elevation in HA in the fetus and neonate compared to in the adult. And the distribution of this was quite surprising. I'm just showing you the example from the sheep because it's larger and easier to see, but we've seen the same thing in mouse and rat, and I'll show you in human as well. The HA is not just uniformly distributed throughout the duct, but it seems to be in a ring surrounding the lumen. And the collagen layer seems to be on the outside. Recall that I showed you earlier in my first talk from the mouse that collagen is deposited, at least in the mouse, it's after birth. It seems in the sheep, it is sort of during the process of development, but it is a later event in the development of the duct than the deposition of hyaluronic acid. And collagen seems to be deposited from the outside rather than from the inside. So that in these normal fetal ducts, you can see a ring of HA around the lumen. And that's shown here. It's the HA is in blue and the collagen is in red and the top figures. And then the lower, the HA is in green and the collagen in red. It's a second harmonic generation imaging of the collagen. So the same thing happens, or we see similar surprising findings after prenatal injury. So in the fetal sheep that undergoes hypoxia, we see a significant increase, roughly a doubling in the HA content of the injured duct. In humans who have babies with BA, when we look at the BA remnants, the extrahepatic bile duct remnants taken at Kasai, in the 5 samples we've been able to look at so far, compared to controls, we also see a significant increase in hyaluronic acid present. And if you look at the photo on the right-hand side, this is one of the remnants. You can see that there is sort of a residua of a lumen marked by the black arrow, and everything around it, all that brown is hyaluronic acid. Whereas the blue, it's not stained for collagen here, but when we've looked otherwise, that is collagen. In adults, we also see an increase in HA with injury. The example here is from patients with PSC, but since the levels start off much lower and the increase is not considered to be significant, although I'm sure if we looked at other samples, we would find it to be. So again, I just want to emphasize that HA is surrounding the lumen and that collagen is on the outside. So fetal hypoxia in the sheep model increases HA content, although when we looked at collagen content, it is not increased. So if you look at this graph on the left, it just shows you the increase in HA in the normal and injured sheep duct. If you look at the images on the right, also taken from the sheep, when we looked at collagen and quantified it, we saw, if anything, a significant decrease in the thickness of the collagen layer, although it appeared to be more dense. With the samples we have thus far, it has not achieved statistical significance, but when we look at that collagen across all our samples, it does appear to be more dense. And so we're doing additional analyses on this collagen. It also looks much more aligned. And so one of the questions we're asking, which we haven't been able to answer yet, but I raise it as a question, is whether this additional HA, which is going to swell, that's what HA does it actually compress the collagen in the duct? Thinking about the other side, does the increase in HA compress the lumen itself or somehow impede bile flow? So we've tried to address this in a couple ways and are continuing to work on additional approaches. But first of all, we looked at using our bile duct on a chip device. We looked at if we made channels through collagen or collagen with either high molecular weight or low molecular weight hyaluronic acid, shown in the middle and the lower rows of photos. We asked whether hydration caused that HA to swell and whether it would have a significant impact on the size of the lumen that we made. And in fact, we saw that with, particularly with low molecular weight HA, we saw really a marked narrowing of that lumen, suggesting that at least conceptually, it would be possible when you had a very large amount of HA deposited to have some narrowing of the lumen. We have looked at these injured sheep, and when we look at their livers, we do see bile plugs, and we also see elevations in serum bilirubin consistent with some kind of obstruction, but we don't have any details on whether the cause is actually HA-mediated swelling or not. But I just show that because it is consistent, if not definitive. I also want to note that we see significant changes in the bile itself in this sheep model. I want to look on the over here on the upper left. We stained these sheep bile ducts for the presence of mucus. This is an Alcian blue stain, and I think it highlights beautifully the increase in peribiliary gland proliferation that is seen in the injured duct. And it also highlights that these injured peribiliary glands are making mucus. So the quantification is shown in this little table just below the images. And you can see that particularly in an animal that has been in the biobag for 21 days, so has been sort of maximally stressed, that there is an increase in mucus-producing peribiliary glands from a baseline of 15 to 17% to 22%. We have also noted in some of these sheep that there are goblet cells along the lining of the mucosa. In other words, cholangiocytes that seem to have undergone some metaplasia, so that they are producing mucin. I don't think that this happens or happens extremely rarely in human BA patients, at least at the time that we see their bile ducts, their extrahepatic bile ducts. But it does suggest that at least in this sheep model, there is increased mucus in the bile. We were able to obtain some bile from these injured fetal sheep. And we're able to carry out viscosity measurements. And what we found was that there was a significant increase in the viscosity of those that had goblet cells. This is work that we need to continue to try to sort out, but it does suggest the possibility that bile itself might be abnormal in the setting of an injured fetal duct, particularly an injured fetal duct, and that might have implications for viscosity and for obstruction. I just show that because I think it's interesting and provocative preliminary data. So again, I just want to highlight that what I've shown you is that there's an increase in hyaluronic acid in the normal and particularly in the injured fetal extrahepatic bile duct, and that suggests the possibility of obstruction. I now want to consider what HA does to the actual cells of the extrahepatic bile duct. So what we did first was we took primary neonatal mouse cholangiocytes and we cultured them as spheroids in either a plain collagen matrigel mixture or with the addition of low molecular weight or high molecular weight hyaluronic acid. And what we found was that in the presence of the high molecular weight HA, cholangiocytes just took off. It was actually very hard to do this because we couldn't, to try to adjust the density of the cells that we cultured because these cells just went so crazy in the high molecular weight HA. So clearly cholangiocyte growth was stimulated by high molecular weight HA. To look at the influence of HA on matrix formation, we took co-cultures of neonatal mouse extrahepatic duct, cholangiocytes, and fibroblasts. So these are primary cells. The cultures are primarily fibroblasts, although there are cholangiocytes present. And we made something called fibroblast-derived matrices, where we cultured these cells either on a control dish or the control dish with the addition of low molecular weight HA or high molecular weight HA. And so if anything, we saw that the fibroblasts actually proliferation decreased, but nonetheless, in the presence of the high molecular weight HA, we saw a dramatic increase in collagen production that's shown here in blue. It's measured by collagen second harmonics generation. It was present and it was quite highly aligned, suggesting that what HA does to fibroblasts in the neonatal duct is it induces the production of collagen and likely the alignment of collagen as well. So I just want to conclude by saying that what I've shown is that fetal extrahepatic bile ducts have increased HA at baseline and after injury. And we know that HA is associated with scarless healing in the fetus from multiple other organ systems. In the injured fetal and neonatal ducts, as well as in Kasai remnants, this HA is in a periluminal ring with an external ring of collagen. And there is some evidence that this HA causes compression of collagen in the one direction, a potential luminal obstruction in the other direction. And as I said, this is associated with massive PBG expansion, certainly in the sheep model. With increased mucus in the bile. And so we think that thinking about this BA from the perspective of fetal wound healing may be useful in the long run as we attempt to sort out what is actually happening in this disease. So as I'd mentioned before, our working model is that increased HA leads to swelling, pressure on fibroblasts and peribiliary glands with increased collagen and cell proliferation. The increased number of PBGs may also increase pressure in that HA layer. There may be viscous bile, and the features of fetal wound healing lead to high potential for obstruction, as shown in the graphic here. So I just want to briefly highlight the ongoing work that we're doing. We are actually, we're collaborating with a group at Wake Forest that can directly measure the molecular weight of HA in the injured neonatal and adult mouse extrahepatic bile duct using nanopore sensing technology, which is pretty amazing. And we're also asking whether what HA is doing is actually causing crowding. We are studying the location of hyaluronidases and are trying to use our bile duct on a chip to better study what HA may be doing in the context of the duct. Finally, we would like to be able to directly assess duct narrowing in the setting of injury. This is a snap frozen extrahepatic bile duct from one of the fetal sheep from the biobag, and it has a relatively narrow lumen, but we've only been able to do this in one duct. And so this is something that we're going to have to find another model to work on. But it will be intriguing to use this snap freezing method to see if in fact there's narrowing in the setting of fetal injury. So finally, I just want to spend 1 minute just putting together the two talks that I've given, suggesting that if we think about biliary atresia as an issue of the fetus, we can think about the fact that the fetal bile duct is susceptible to injury, and it's susceptible to the spread of that injury and to obstruction, and that this all occurs in the context of a fetal wound healing program. After birth, there is a switch to adult wound healing. It is a gradual switch, but nonetheless, it clearly occurs. And also at this time, there are fibrogenic cells in the submucosa. They're primed for fibrosis, and both the mechanical features of the extrahepatic bile duct, as well as the potential for swelling of HA after injury, may lead to increased fibrosis from these primed cells. And so finally, I just want to end with this one slide, asking the question of what happens at birth. Does something significant change around birth, and is it important to biliary atresia? So one thing we know is that around the time of birth, and birth is sort of, I'm using that roughly, this could be the third trimester as well, or the neonatal period, bile flow increases markedly, and that leads to the potential for obstruction, for cholestasis, and for leakage of bile into the extrahepatic bile duct submucosa with associated damage. There's also oxidative stress at birth, and peribiliary glands switch from glycolytic to oxidative metabolism, and that may be important in leading to increases in PBG proliferation and repair during this time period, may also lead to increased damage, as you might expect from oxidative stress. And finally, as I've mentioned before, there is this switch to adult wound healing. And in the context of fibrogenic cells in the duct, it is very likely that there's increased deposition of collagen 1. In fact, it's certain that there's increased deposition of collagen 1 at this time. It may also have a potential impact, the collagen 1 might have a potential impact on peribiliary gland behavior. But these are questions that I think we need to start considering experimentally. So I finally want to acknowledge again the people who did this work. Iris De Jong did the majority of the work in this particular talk, and others in the group, including Yu Du, who does the bile duct on a chip, Dongning Chen, who does the fibroblast-derived matrices, as well as Jess Llewellyn, Kapish Gupta, and Cici Chen, have all contributed. And I would be remiss if I didn't note that Bill Gaynor and the biobag group, including Meredith Gunder and Mallory Hunt, were really essential to carrying out this work. So thank you very much. I'm happy to take questions.
Thank you very much again, Dr. Wells. So we have a question here from the audience. Dr. Alfredo Santamaria is asking, could there be any difference between this model of bile duct atresia in mice and chimps with that of the zebrafish model, in which there are apparently interesting findings in cytochrome expression and other findings?
So that is hard to say. I think it depends on what the specific differences, what the specific findings are that you're referring to. So there is no question in my mind that there are some significant differences between the zebrafish extrahepatic bile ducts and the mammalian extrahepatic bile duct. Certainly some things are very similar. We have published a toxin-based model of extrahepatic bile duct injury, where there seem to be marked similarities in the zebrafish and in mammalian ducts. But clearly that is not true for every toxin. It is not true for every finding. So I don't know I don't know specifically what data you're referring to, but I can say that there are both similarities and differences. Some of the work that we've been doing recently, we have looked at the zebrafish for in one or two experiments, and we've seen some differences. So yeah, I think the sheep and the mouse and rat are different enough from human for us at this point.
Paul, another question for Dr. Wells from the audience. What do we know about the compilation of circulating growth factors in the blood milieu of early infancy, and if they may have a role in the fibro-obliterative process of VA?
Right. So in early infancy, I think others here are more knowledgeable about that than I am. I do know that there can be high levels of TGF-beta, which is highly fibrogenic, and that almost certainly acts on fibroblasts of the extrahepatic bile duct. Some of Jess Llewellyn's work in my lab does suggest that the fetal versus the adult fibroblasts also have quite different mechano responses. And so it's hard to know from work that we have done on stellate cells. TGF-beta and other soluble factors certainly interact with the stiffness of the mechanical environment as far as fibrogenesis. So it would really depend on, have to look at the specific cell type and look at specific growth factors to sort out whether that's actually relevant in the fibrogenesis by those cells, if that answers the question.
Dr. Becerra has also a question.
Right, I saw he'd asked about what is the source of HA within the ducts. So we have looked at the fetal duct, and we do see the presence of hyaluronic acid synthases 1, 2, and 3. Those are the major ones, and they're all present at seemingly high levels in the fetal duct. They are present primarily in cholangiocytes, but we do see some in the submucosa as well. So the antibodies for the hyaluronic acid synthases are quite controversial. So we're confirming all those data with real-time PCR and with other methods. But there's no question that these are present in the ducts and seem to be present at quite high levels. And I think it's cholangiocytes.
Becky, I thought that fibroblasts in smooth muscle cells also common sources of HA, and if so, it looks like you might be. It looks like you're already considering populating your chip model by adding some fibroblasts and maybe different concentrations to see if that matters.
Absolutely. Yeah. So certainly HA, we do see it produced by fibroblasts as well. It's just, there are more cholangiocytes and it's just much more impressive, but it is produced by both populations. And yes, UDO in my group has made he's made a number of different bile ducts on a chip, including this lovely 2-channel chip that has a vascular channel and a biliary channel with fibroblasts in between. These are all human cells that he's used. He's been able to show that that he can actually get transmigration of immune cells across the vascular endothelium towards the bile duct, and is doing a lot of work, quite a bit of work with that. But one of our interesting questions is, what happens if we put HA into the matrix of that, that device? What happens to cholangiocytes? And what happens to fibroblasts, both in the presence of HA and also with other growth factors, other injury agents, other permutations. So that's work is ongoing. Yeah.
So, Dr. Waltz, you mentioned that you have to plan to quantify the particular molecular weight of hyaluronan in fibrotic biliary tract. Is that correct? You have that data?
Yeah, so that's it turns out that determining the size of a given population of HA is much easier said than done. And it can be done by sort of a gel filtration method, but you obviously need a huge amount. I mean, you simply cannot do that with a neonatal mouse bile duct, as I'm sure you can imagine. But this scientist, Adam West at Wake Forest, has developed this nanopore technology that can actually categorize HA into high and low molecular weight and everything in between, using nanogram amounts of HA. And so we are collaborating with him. We're just sending off to him our first batch of samples. We know that with something like 10 neonatal mouse extrahepatic ducts, we should be able to get an answer. So I think that will be very interesting. I strongly suspect that we're going to see very high molecular weight HA in the neonate and less so in the adult. But it'll be interesting to see. Just don't know yet.
Thanks.
Thank you very much. We are on time, and you can save your question for the Meet the Expert session. So now we are going to go to session 3. So thank you very much again for our speakers, and thank you, Dr. Masolini.